Okay, so we're going to start chapter 5. We're going to talk about the molecules of life. Specifically, we're going to look at these four. We're going to look at carbohydrates, proteins, lipids, and nucleic acids. Some textbooks out there are adding a fifth group. They're adding ATP in that group. Our textbook does not. I don't have an opinion on whether that's right or wrong. I mention it only because if you have a different version of the textbook, or you come across one later, or you have an instructor who insists upon a fifth group, what they're probably looking for is ATP. We're going to look at things like polymers. We're going to define a polymer in our next slide, and monomers. And then you're going to want to know those. You're also going to want to know what these macromolecules do with regard to life, what their function is for cells or even just within organisms. So let's just start off with an overview. Same as we did last time. So all living things are made up of four classes of biological molecules. We're made up of carbohydrates, lipids, proteins, and nucleic acids. If we put a definition, an official definition on macromolecules, macro is large, right? So we're going to talk about large molecules that are composed of thousands of covalently bound atoms. So the key to macromolecules is that these bonds are covalent. We are not talking about ionic bonds like we do in the ionic salts. Most of the macromolecules that we'll talk about are polymers. Polymers are large molecules that contain repeating subunits, many repeating subunits that again are covalently linked together. Those subunits are called monomers or building blocks. So monomers covalently bound together make up polymers. The same way that if you can picture a train in your head, and I don't necessarily want you to picture the engine or the caboose, I want you to picture a whole string of boxcars. You can make your train very long or you can make your train very short. But individually, each of those boxcars is considered a monomer. It's a subunit. And when I string all of those monomers or those boxcars together and I click them together, what I end up getting is a very long chain of monomers. And that long chain of linked monomers is called a polymer. Down here at the bottom, we have some just basic terms that you should know. Polys are going to be many. Di will be two. Mono will be one. Meros will be part. Tri will be three. Some base stuff that you probably already know, but just make sure that you've got that in the back of your mind as we're going through. And remember too that this structure and function thing is inseparable. We touched on that in chapter 1. We hit it again in chapter 3. Here we are in chapter 5, and we're saying it again, and we're going to say it again in the future. Structure and function are inseparable when we talk about biology. So again, polymers, long molecules made up of many similar, smaller building blocks. Those similar, smaller building blocks are called monomers. When we talk about 3 of the 4 macromolecules, they are considered polymers: carbohydrates, proteins, and nucleic acids. Notice that lipids are not on this list. We'll talk about them very specifically in a little bit, but they don't actually fall into that polymer category. They have their own category going on. So carbohydrates, proteins, and nucleic acids are all going to be considered polymers. In that same vein, we need to talk about polymer construction or polymer deconstruction. Am I building up? Am I constructing a polymer, or am I breaking it down into its smaller subunits? So if I'm constructing a polymer, if I'm putting together smaller subunits, if I'm joining monomers, that process is an anabolic process or anabolism. And that occurs through a very specific type of reaction. It occurs through a condensation reaction, also called a dehydration synthesis reaction, sometimes even just called a synthesis reaction. But if I'm constructing a polymer from its smaller subunits, its monomers, then that's going to happen through a condensation or a dehydration synthesis reaction. If I'm deconstructing or breaking down, taking apart a large polymer and breaking it into its smaller subunits, I'm going to do that through a hydrolysis reaction. Okay, so I'm going to deconstruct it. That's called a catabolic process, or it's called catabolism. And we're taking a more complex molecule and we're breaking it into its smaller subparts. Okay, so let's just look at, let's look at both of those really quickly, and then we'll break on this video and pick up with our next. But we want to have a synthesis or a breakdown of polymers. So remember that condensation reactions, more specifically called dehydration reactions, occur when I bring two monomers together, and in so doing, what we're going to do is we're going to release a water molecule. Okay. So that's called a condensation reaction. The opposite, I'm going to skip down to the third bullet, the opposite can also happen. I can take a large polymer and I can disassemble it. I can break it into smaller monomers, also using water. So water plays a role both in condensation and in hydrolysis reactions, but I'm going to use that water to essentially break apart the bonds and create two smaller monomers. A very interesting type of protein called an enzyme. So enzymes are proteins. Enzymes are macromolecules that are going to speed up a dehydration reaction. They're going to speed up a synthesis reaction. Here's a visual for those of you who are like me and need that visual. We've got this slide and the next. So this is an example of a dehydration reaction where I have this short polymer and I have another short, this one's a monomer. It could have been a polymer, but in this image it's a monomer. And what I'm going to do is the hydrogen off of this short polymer and the hydroxide off of this short polymer are actually going to get pulled out. We're going to form water, and we're actually going to form a bond between this monomer here and the number 3 of the original polymer, and I'm going to get this longer polymer right here. So remember, dehydration reactions are going to require energy. So I need energy to break these bonds and allow the new bond to form here. They require energy and they are often aided by enzymes. Okay. Our next, sorry about that, our next is a hydrolysis reaction. So in a hydrolysis reaction, I have this nice long polymer, the one that I just formed in the slide before. I have this nice long polymer, and I want to break part of it off. I want to take it to a shorter polymer and a monomer. And what we're going to do is we're going to use water. So water is actually going to come in here and it's going to break this bond right here. It's going to break this bond, and what we're going to end up with are this shorter polymer over here and a monomer over here, and that's how we're going to create a hydrolysis reaction. So hydrolysis breaks a bond and uses water to separate a longer polymer into a shorter one. We'll see hydrolysis reactions are going to dominate the digestive process. They're guarded by very specific enzymes, and it's a big chunk of what just is digestion for all mammals. So that's where I'm going to stop this video. We'll pick up with diversity in polymers. I think our next slide up is going to be a whiteboard slide, so kind of be thinking about what we just talked about, and we'll see how you do on that.